Hi everybody, this is going to be a review for our Microevolution lab for our Bio 1 course at Mount San Antonio College. So let's start off first by talking about our antibiotic resistant bacteria activity. We start off with our petri dish, which is meant to represent our body, and in our body we're going to have this infection of bacteria. And we're going to start off with 13 brown beans and 6 of the red beans and 1 black bean. And that's going to be our starting population of our bacterial infection in our body. And the idea is that the brown beans are the easiest to kill with the antibiotic if you take it. The red beans are resistant a little bit to the antibiotic, and the black beans are the most resistant and therefore the hardest to kill. All right, so I'm going to roll my dice, and based on what I roll, if I roll a 1, 3, 5, or 6, that means I remember to take my antibiotic. And when I take my antibiotic, that means that I'm going to kill 5 bacteria, and we're going to always start with the brown bacteria first. You can't kill any of the other bacteria till you get rid of the brown. And so I get rid of those, and then for every color that survives, I'm going to add one. So I'm going to add, in this case, 1 brown, 1 red, and 1 black. And I'm going to want to note this new population on this chart so that I can go on to round 2. Okay, so my data should look like this, where I should now have 9 brown bacteria, 7 of the red, 2 of the black. So I roll the dice again. I roll a 6, so once again I'm going to remove 5 because I took my antibiotic, and I'm always going to remove the brown ones first because they are the least resistant to the antibiotic. And then for every color that survives, I'm going to add 1. So I'm going to add 1 to the red, 1 to the brown, and 1 to the black. Okay, so we're going to add that, and then once again we're going to record our data, and now you'll see we have 5 of the brown bacteria, 8 of the red, and 3 of the black. And you notice that our population is going down, which is a good sign. And so from here on out, we continue to roll the dice and continue the same process. I'm going to speed this up, and you can record your data or review your data because it's basically the same process as you go. And we're going to keep doing this until we either get rid of all the bacteria in our dish or the black bean population reaches 12, at which point the infection becomes out of control and is no longer treatable with this antibiotic. Okay, for our next activity, we're going to use plastic ants to represent genetic drift and gene flow. And we're going to start off with 4 brown ants, 8 black ants, and 8 black ants with a dot on them that are going to be our heterozygote ants. So our brown ants are going to be homozygous recessive, little b, little b. Our black ants are going to be homozygous dominant, big B, and our heterozygotes are going to be big B, little b, and we're going to mix those up. And the first thing we're going to do is 4 of our ants are going to be crushed by a rock just from bad luck, and so we're going to remove those from our population. We're going to take the remaining ants and we're going to pick 3 pairs of 2. They're going to represent the breeding pair, and that's what you see here. And then using this chart, we're going to, based on the breeding pairs, determine 5 offspring for each breeding pair. And that's going to be generation number 1, or essentially the babies of those breeding pair. So I'm going to roll the dice each time to determine what my offspring for each pair will look like. Okay, so if I speed that up, this is my first generation right here, the 15 offspring from my original pair. So these 15 ants represent generation 1 and go on this chart, and I can use those individuals to calculate the frequency percentage of alleles of the dominant and recessive. And then using that pile of ants, I do the same thing. I pick 4 out that get hit by a rock, and then I pick 3 breeding pairs from that pile using the generation 1 ants, and I do the same thing. I go through and I figure out the 5 offspring that each of those breeding pairs have, and that becomes generation 2, and I can use generation 2 then to also calculate allele frequency and see if that is different from my starting population. To demonstrate gene flow, what we're going to do is we're going to take a population of 10 all black homozygous dominant ants, and from our generation 2 we're going to pick 4 ants from our pile and add that to those 10 and calculate the allele frequency difference and see if that changes. For our next activity, we're going to do a natural selection predator-prey model, and for this we will use these different colored chopsticks that are different lengths, and these are meant to represent birds or some other animal. I particularly like to say they're birds with different bill lengths, and what you're going to do is we're going to take some brown ants, 20 brown ants and 20 black ants, and we're going to take our ants and spread them out. And we want to make sure we don't use the heterozygous ants. In this case, we're just using the black and the brown, not the ones with the dot on it. So we're going to take our ants, spread it out over the habitat cloth, and then based on what your instructor tells you, I typically do 20-25 seconds. You're going to try to get the ants, pick them up one by one, and put them in your stomach, and that will be the end of round 1. Now we're going to record our data here, so each person is going to count how many ants they have in their stomach, and they're going to share that information, and we're going to fill out this chart here that shows how many of each type of being each predator got, and we're going to total that number up, and then for every survivor, for everyone that survived, we're going to add 1 offspring, and this will then be the start of generation number 2. We do the whole thing again, and then we do it one more time for a total of 3 generations.